You will conduct observations of potassium permanganate solid in water at 5 degrees Celsius, 25 degrees Celsius, and 40 degrees Celsius. Caution: the potassium permanganate crystals used in this experiment make a very powerful dye that can permanently stain. Wear gloves and be careful not to spill chemicals. Gather 3 clean, dry test tubes. The test tubes must be perfectly dry for this exercise. Locate a ruler and a felt tip marker or wax marking pencil. Place a mark 7 centimeters from the base of each test tube. Place the test tubes in an empty cup, beaker, or 24 well plate. Transfer a small amount of potassium permanganate crystals to a scrap of paper. Use the forceps provided in your dissecting kit to transfer 5 crystals to each of the test tubes. Label 3 short clear plastic cups cold, ambient, and warm. To the cold cup, add 2 to 3 ice cubes or fill the cup 1/3 full with crushed ice. Add tap water until the cup is 3/4 full. Let the water equilibrate for 5 minutes. Use the thermometer to measure the cold water temperature and record it in data table 2. The ice water temperature should be close to 5 degrees Celsius. Fill the ambient cup 3/4 full of tap water that is within a few degrees of 25 degrees Celsius. Use a thermometer to measure the exact temperature and record in data table 2. Fill the warm cup 3/4 full of tap water that is within a few degrees of 40 degrees Celsius. Use a thermometer to measure the exact temperature and record in data table 2. Boiling tap water is 100 degrees Celsius. Warm water, 40 degrees Celsius, can be obtained from most faucets without additional heating. Use a short stem pipette to slowly add water from the cold cup to the first test tube. Tilt the test tube so that it rests at a slight angle, and the water may gently flow down the side of the test tube. There will be some diffusion when the water contacts the crystals. Fill the test tube to the line. Observe the solution in the test tube and record observations at 0 minutes in data table 2. Very gently place the test tube into the cold cup. Make certain that the tube base is fully in the solution and that no water can enter the test tube. Repeat the preceding steps using the ambient water and the warm water in place of the cold water. After 5 minutes, observe the solution in each test tube. Compare the test tubes relative to one another. Record observations in data table 2 under Observation after 5 minutes. To clean the equipment, pour the potassium permanganate solutions down the sink and flush with copious amounts of tap water. Wash the cups and test tubes with dish soap and tap water and dry with paper towels. Dispose of the used short stem pipette in a trash bin. Return equipment to your kit for future use.